The human circulatory system is a complex network of blood vessels that supplies oxygen and nutrients, removes wastes, and transports immune and hormonal cells and factors. The development of vessels, or angiogenesis, is exquisitely controlled by signals and pathways from adjacent tissues that the vessels serve. This allows rapid response to environmental stimuli such as hypoxia and nutritional and metabolic stresses. Not all of these molecular signals and pathways are well understood. To identify genes involved in angiogenesis, researchers studied zebrafish mutants with fluorescently labeled blood vessels. They found that the loss of trionyl tRNA synthetase, or TARS, or isoleucyl tRNA synthetase, or IRs, led to excess trunk and cranial angiogenesis, and that excessive angiogenesis in the mutants could be rescued by injecting wild-type copies of TARS and IRs. Aminoacyl tRNA synthetases are known for their primary function in accurate protein synthesis and play an important role in the unfolded protein response, or UPR pathway. Some aminoacyl tRNA synthetases, however, are also known for additional functions such as regulation of angiogenesis. This study is the first to identify the role of two new aminoacyl tRNA synthetases, TARS and IRs, in promoting angiogenesis. To understand the mechanism for excessive angiogenesis in TARS or IRs mutant zebrafish, researchers studied the expression of genes involved in the UPR pathway. While it is known that the UPR genes such as ATF4, ATF6, and XBP1 are upregulated in response to endoplasmic reticulum, or ER, stress, this study found that loss of TARS or IR's function not only resulted in upregulation of these genes, but also increased expression of the angiogenic growth factor VEGF. The induction of VEGF was significantly reduced by the knockdown of ATF4. This suggests that UPR pathway induction is necessary for both VEGF expression and increased angiogenesis in TARS-deficient zebrafish. These observations suggest that loss of synthetase function as a result of nutrient depletion, genetic mutation, or small molecule inhibition produces ER stress, followed by increased ATF4 and VEGF levels that in turn promotes vascular branching. Although transfer RNA synthetases have been shown to be involved in angiogenic regulation through non-primary mechanisms, this is the first evidence showing that disruption of the primary function of transfer RNA synthetase genes can cause excess angiogenesis through the activation of UPR pathways. This study also highlights the usefulness of the developing zebrafish as an in vivo model for studying metabolic regulation of angiogenesis.